Hello and welcome to Zoology 142. This is our second lecture on the circulatory system. Today we're going to be looking at the heart, and the heart is a muscular pump that is responsible for pumping blood all throughout the body, and as this slide says, it does a tremendous amount of work. It will pump over 1 million gallons of blood a year through over 60,000 miles of blood vessels, and it does this for your entire lifetime without even resting. It really is a miraculous organ. Okay, we need to talk a little bit about the area where the heart is located. Most of you know the heart is beneath the ribs. It's actually in an area called the mediastinum or mediastinum, and this is the technical name for the area between the sternum and the vertebral column. And so the heart itself is actually sandwiched between the left and right lungs, and so in order to visualize it, sometimes heart surgeons actually have to retract the lungs laterally to be able to view the entire heart. So again, the heart is in the mediastinum. The heart is also covered by a protective sac called the pericardium. Remember, peri means outside or around, and cardium means heart. The pericardium is formed by two different layers. The first of these layers are the fibrous pericardium. The fibrous pericardium is composed of dense connective tissue, and its function is to protect and anchor the heart to the surrounding structures. It also prevents the heart from overexpanding or overstretching. The second main layer of the heart is something called the serous pericardium. The serous pericardium is actually a thin double membrane that encloses pericardial fluid. The outer layer is called the parietal layer, and the inner layer is called the visceral layer. These two layers have sandwiched between them the pericardial cavity, and the pericardial cavity is filled with a little bit of pericardial fluid. And the function of this fluid is to lubricate the heart, basically prevent friction between the beating heart and the surrounding organs. So normally there's just a few milliliters of this serous fluid in between the pericardial layers. But in certain circumstances, the pericardium can become engorged with fluid or even blood. So we talked about cardiac tamponade, if you remember from medical terminology, and this is a condition where we have excessive amounts of fluid building up in that sac, and as a result the heart cannot contract or relax properly, so it starts pumping less and less blood. And so the cardiac tamponade is a very life-threatening condition, and the only way to cure it is basically to pierce the pericardial sac and draw out that fluid with a large syringe. Okay, so now that we've removed the pericardial sac, let's take a look at the layers of the heart wall. The first of these is the outer epicardium. Remember, epi means outside and cardium means heart. So the epicardium is actually the same layer as the visceral pericardium that we talked about in the previous slide. Now the myocardium, the second layer, is deep to the epicardium. The myocardium is made up of cardiac muscle tissue as well as some connective tissue. And the myocardium is the thickest layer of the heart and the one that's responsible for its contractile force. Finally, the endocardium is the deepest layer within the heart, and it's composed of squamous epithelial tissue as well as some connective tissue. And the purpose of this endocardium is to allow a smooth, friction-free membrane over which blood can flow. Remember from the previous chapter we talked about what happens when blood flows over a rough surface is that the platelets can be activated, causing intravascular clotting. And obviously you don't want clotting inside of unbroken blood vessels, and by the same token you don't want clotting inside of your heart. And so we have this very smooth endocardial layer made up primarily of epithelial tissue and a little bit of connective tissue. Now let's take a look at the external anatomy of the heart. On the outside of the heart you can see these small ear-like flaps which are called auricles. The auricles are actually the external parts of the atria or top chambers of the heart. They look like ears, and so we call them auricle because auricle means ear. Now you might be looking at the left and right and saying, well, he's got this backwards, but here's an important point. When we're referring to heart anatomy, we refer to this heart as if it were your own heart. So imagine that you are facing yourself, that you're sitting on the screen looking back at yourself, that your left auricle would be on your left side and right auricle on your right side. So the heart apex, the tip, always points to the left side of the body. So if you're ever confused what side of the heart you're looking at, the pointy part is the left-hand side. So below the auricles are the ventricles. There are two ventricles, a left ventricle and a right ventricle, and they cannot be easily distinguished from the outside. The left ventricle tends to be the larger of the two because it has more work to do, and we'll talk more about that in the upcoming slides. And as I said before, the apex of the heart is the pointy part of the heart, and it usually points to the left-hand side, whereas the base is the flat part that tends to sit on the diaphragm. So now that we've taken a look at the major external structures of the heart, let's take a look at the blood vessels that lead into and out of the heart. The first of these blood vessels is the vena cava. The vena cava is the largest vein in the body, and it brings blood back from the superior area of the body, that is the head and the neck, or the inferior area of the body, that is the trunk, the thorax, and so on. And all this blood is routed directly to the right atrium. The next blood vessel we have is the pulmonary artery. So let's look at the two words here. Pulmonary means lungs, so this is a blood vessel that must lead to the lungs, and artery is a vessel that leads away from the heart. So this is a vessel that is transmitting blood from the right ventricle to the lungs, the pulmonary artery. And here we have the pulmonary veins. Remember, pulmonary means lungs, and here vein is a vessel that carries blood back to the heart. So the pulmonary veins takes the blood from the pulmonary capillaries and brings it back to the left atrium of the heart. Okay, the aorta is an artery, and arteries carry blood away from the heart. And the aorta is probably the largest diameter artery in the body because it carries blood from the left ventricle to the rest of the body. And there's different parts of the aorta. The part coming just out of the heart is going to be the ascending aorta, and then the candy cane part we call the arch of the aorta, and then finally it will start to descend, and then we call that the descending aorta. So the aorta carries oxygen-rich, high-pressure blood to the tissues of the body. We call these tissues the systemic circuit. And finally, the smallest blood vessels visible on the heart are the coronary blood vessels. Particularly important are the coronary arteries. The coronary arteries are the ones that actually provide the myocardium of the heart with oxygen-rich blood. It seems kind of counterintuitive. You see all these very large blood vessels going in and out of the heart, but none of these actually supply the heart muscle with oxygen. It's actually these little bitty Manini, tiny little coronary vessels that supply the heart with oxygen. And because of their small size, we can often get blockages or clots that can cause serious trouble. So as I was saying, the coronary arteries are extremely important because they supply the heart muscle, the myocardium, with the oxygen-rich blood it needs to continue to contract. And so if we have any kind of blockage in these blood vessels, it can very quickly lead to ischemia of the tissues that those vessels are supplying. So in particular, we're very worried about coronary artery disease, or CAD. And coronary artery disease can be caused by a variety of factors, including just spasms of the blood vessels, as well as deposit of fatty material and sclerification of the walls of these blood vessels. So here you can see a cross-section or transverse section of a coronary artery with a large arterial sclerotic plaque. And you can see that at least 1/3 of the lumen of this blood vessel is completely blocked by this plaque. And so as a result, we have less perfusion of the tissues downstream of this clot. So we're going to talk about So in the next slide, we're going to talk about some of the dangers of coronary artery disease. And after that, we're also going to talk about some possible treatments. Of course, probably the biggest danger of an obstructed coronary artery is something called a myocardial infarction, or MI. This is otherwise known as a heart attack, and it's usually caused by a blockage of the coronary arteries. When we have a heart attack, we actually have the death of many of these myocardial cells. And the important thing to realize is if you survive that heart attack, those dead cells are not replaced with new muscle cells, they're replaced with scar tissue. And as a result, the heart has less contractile force than it did before. Myocardial infarcts tend to be most serious when they occur in the left ventricle, because as we'll learn in a few slides, the left ventricle is the one that's responsible for pumping blood to the entire body. And so if that's damaged, it can be very difficult to recover from an MI affecting the left ventricle. So as I was saying, coronary artery disease is one of the primary causes of heart attacks or myocardial infarctions. So one way to prevent or remedy coronary artery disease is to do something called a bypass graft. Bypass grafts are done using open heart surgery, so we actually have to open up the chest, put the patient sometimes on a bypass machine, and here we're taking a vessel from the leg and we're stitching it onto the aorta, which has a nice high pressure oxygen rich blood supply, and then we're routing that blood vessel downstream of an obstruction in the coronary vessels. So basically it's called a bypass graft because we're bypassing blood around the obstruction. We're not removing the obstruction, but just bypassing blood around it so that we can maintain adequate blood flows to the tissues downstream of that obstruction. Now, you've probably heard of a single bypass, a double bypass, or even a triple or quadruple bypass. So it's possible to perform this operation up to 4 times to divert around up to 4 different blockages in the coronary blood vessels. Big picture here is that bypass operations use blood vessels from the legs to divert around obstructions in the coronary arteries, and that the bypass graft surgery is a major inpatient surgery. A less invasive alternative to a coronary artery bypass graft is something called balloon angioplasty. Remember that angio means vessel and plasty means surgical repair of. So during this procedure we actually insert a small catheter in the blood vessels of your thigh, and we thread that catheter all the way up to your coronary arteries. And then we inflate a little balloon at the tip of that catheter, and we use it basically to just squeeze the fatty deposits in that artery over to the sides so we can give ourselves a nice round wide lumen through which blood can flow. So balloon angioplasty has the added benefit in that we can reduce a blockage in a blood vessel without having to go through major open heart surgery. Unfortunately, balloon angioplasty isn't always a permanent procedure. Oftentimes the plaque in that artery can begin to close back up in the successive months and years, and so oftentimes we will do a balloon angioplasty in conjunction with something called a coronary stent. A stent is basically just a little wire cage that's threaded around that balloon, and when the balloon is inflated, the cage expands and helps to hold the blood vessel open. So the balloon is threaded out and deflated, and the cage or stent remains behind to hold the blood vessel open. And so stents were thought to be a more permanent solution to this obstruction in the coronary artery. Okay, now we're going to learn about the mechanics of the heart. And so the heart is a 4-chambered pump, and the 2 top chambers are called the atria, or atrium, which is singular, and their job is to pump blood into the ventricles, which are the lower chambers. So they really aren't very muscular because they only have to pump blood a very short distance. The ventricles, on the other hand, have very muscular walls, and they're divided by something called the intraventricular septum. They are the most muscular chambers of the heart because they have to pump blood through lots and lots of blood vessels. For example, the right ventricle has to pump blood from the pulmonary artery to the lungs, the capillaries in the lungs, and then bring it all the way back to the heart into the left atrium. On the other hand, the left ventricle has to pump to what we call the systemic circuit, which is basically the remainder of blood vessels in the rest of the body. So it tends to be the most muscular chamber in the heart. Now, the heart chambers are separated by one-way valves. And why do you think this is necessary? Well, having one-way valves prevents the mixing of blood from oxygen-rich and oxygen-poor areas of the heart, and it also prevents blood from going backwards, which would be very, very inefficient. So the first of these valves we have are the AV valves, or atrioventricular valves. As the name implies, these valves separate the atria from the ventricles. There are 2 different atrioventricular valves. The first of these is the tricuspid valve. The tricuspid separates the right atrium from the right ventricle. One good way to remember this is that tri and right both have an R in it. On the other hand, the bicuspid or mitral valve separates the left atrium from the left ventricle. It's called the mitral valve because if you look at it in the right way, it kind of looks like a pope's hat, which is called a mitre. But the good way to remember which one is which is that the tricuspid has an R in it and tricuspid is on the right side, so that must put the bicuspid on the left side. Now the tricuspid and bicuspid are both anchored to the heart wall by something called chordae tendineae. Literally, these are your heartstrings. The functions of these heartstrings, however, have nothing to do with love or affection, but their function is to prevent the valve from prolapsing or going backwards when the ventricles contract. And so they're basically fibrous connective tissue ropes that keep the valve only opening in one direction. And they're anchored to the heart wall by something called papillary muscles. Now the papillary muscles keep tension on the chordae tendineae, but they don't actually mechanically open or close the valves. We're going to see that that's more of a process of the atria contracting while the ventricles are relaxing. So big picture here is the chordae tendineae help to support the valves and prevent them from prolapsing or blowing back the wrong direction. So whereas the atrioventricular valves separated the atrium from the ventricle, these semilunar valves help to prevent backflow from the blood vessels leaving the heart into the ventricles. So we have two of these. The first one is called the pulmonic semilunar valve, and pulmonic is referring here to the pulmonary artery. So take a look at the pulmonic semilunar valve. So imagine what happens when the ventricles contract. It's going to force blood up into the pulmonary artery, but then the ventricle is going to relax. What would blood do without the pulmonic valve? Due to the force of gravity, blood would tend to just drip right back down into the ventricle, and that wouldn't be very efficient. So the pulmonic semilunar valve keeps blood from backflowing into the ventricles while the heart is relaxing. We have the same thing for the aortic semilunar valve, located here. Again, when the heart contracts, blood is forced from the left ventricle up into the aorta. But when the heart relaxes, that blood has a tendency to go backwards, and it's the aortic valve, or aortic semilunar valve, that keeps it from dripping back down into the left ventricle. Okay, this picture just shows a superior view of the ventricle with the atria removed, and the whole purpose here is so you can see what the valves look like, so you get a better idea of why we call them the names that we do. So first let's look at the semilunar valves. We call them semilunar because they look kind of like half moons. We have the pulmonary semilunar, again, that goes between the right ventricle and the pulmonary artery, and the aortic semilunar, which goes between the left ventricle and the aorta. And then we have the tricuspid and bicuspid. Remember, these were our atrioventricular valves. So on the left you can see the bicuspid, and that's called the bicuspid or mitral because it only has two cusps. On the other hand, take a look at the tricuspid. Here we see three different flaps here. That's why we call it tricuspid. Remember that tri is on the right, and so the tricuspid valve separates the right ventricle from the right atrium. So let's take a look at some heart valve disorders. The first of these is called stenosis. Stenosis just means narrowing of. So we can have stenosis within a valve or stenosis within a blood vessel. And narrowing is going to restrict the flow. And so we can repair this using something called valvuloplasty, or surgical repair of a valve, or we can use a replacement valve. We can get a cadaver valve, we can even use a pig valve. And it's very important that the valves function properly, otherwise we're going to get a really big reduction in the efficiency of the heart. Our next disorder is something called valvular insufficiency or valvular incompetence, and the name should give you a clue as to what this term means. If you think about an incompetent person, it's somebody that's not doing their job correctly. And so here, an incompetent valve is a valve that's not closing all the way when it's supposed to. For example, here you can see the semilunar valves that should ordinarily close when the heart is relaxing to prevent backflow of blood from the aorta back into the left ventricle. On the other hand, if you have an incompetent valve, that valve stays open. So again, a little bit of that blood goes backwards, and so the heart has to basically redo the work that it just did, pumping blood from the left ventricle up into the aorta. And that is valvular incompetence or insufficiency. So now that we've learned a little bit about the internal anatomy of the heart, as well as the blood vessels that lead into and out of it, we need to talk about the job that the heart does. And that main job is to pump blood throughout the two circuits of the body. The first of these circuits is called the pulmonary circuit. Pulmonary here refers to lungs, and circuit refers to circle. We call it a circle because in the pulmonary circuit, the right side of the heart pumps deoxygenated blood from the heart to the capillaries of the lungs and then back to the heart. So the pulmonary circuit begins in the right atrium and ends when blood enters the left ventricle. Why did we go to the lungs? The purpose of going to the lungs was to oxygenate the blood. The second circuit of the body is the systemic circuit. Whereas the pulmonary circuit were just the capillary beds that are in the lungs, the systemic circuit has to pump to all the remaining capillary beds in the body. So it's a much larger job. And this is accomplished by the left side of the heart. The left side of the heart contains oxygen-rich blood that's being pumped to the systemic capillaries. And blood is drained from the systemic capillaries by our vena cava, and that brings us all the way back to the beginning of the loop, which is our right atrium. So if you remember, we said that the left ventricle's job was to pump to the systemic circuit, whereas the right ventricle's job was to pump to the pulmonary circuit. Now, which of these two circuits is larger? If you said the systemic circuit, you're correct. The pulmonary circuit is a relatively short circuit. It goes from the right ventricle to the pulmonary artery, to the lungs, the pulmonary capillaries, back through the pulmonary veins, and so it doesn't have to pump quite as hard as we do for the systemic circuit. As a result, the right ventricle tends to be fairly thin-walled, not very muscular, whereas the left ventricle is incredibly muscular. And this is because the left ventricle has to pump through all the remaining capillary beds in the body. And think about those thousands and thousands of miles of blood vessels. So most of the work of the heart is in fact done by the left ventricle, and that's why it's so thick. Now, one of the big questions you should be able to answer after the end of this lecture is, how does the blood pressure and degree of oxygenation change throughout the pulmonary and systemic circuits of the circulatory system? In order to do this, you can't just study a diagram. You have to be able to draw these circuits as well as the heart chambers and diagram how pressure changes and how oxygen changes throughout the circuit. So now we're going to take a brief break from lecture and talk about how to correctly draw the heart and diagram the circulation through the pulmonary and systemic circuits. I should tell you that I am not an artist, and so drawing a heart like we see it left, it would be impossible for me. So I'm going to give you the idiot's guide to drawing a very simple heart and diagramming the blood flow through those two circuits, the pulmonary circuit and the systemic circuit. So even though I'm going to show you how to draw and diagram the heart, you need to be able to do this on your own. You should do it on your own. So I'm going to give you some incentive to actually stop what you're doing right now, go get a piece of paper, some colored pencils, and come back and we'll draw this together. So if you scan or photograph your drawing, you can upload it to Dropbox on the LaLima website, and if you upload that within 1 week of this lecture, I will give you an extra 5 points on your quiz. Not only will it give you a few extra points, but being able to draw and diagram blood flow throughout the circulatory system will be important not just in this chapter, but also in the chapter on the respiratory system. And so being able to correctly draw this diagram is going to help you on the first exam, on the second exam, and probably 2 or 3 different quizzes that we're going to have. So make sure you can do it. Okay, so Bob Ross here. Today we're going to draw the human heart. And before we do that, we need to clear our mind. Blank mind, blank paper. The first thing we need to do to draw our heart is to draw a circle. This is a happy circle. We will then divide that circle in half using something called the intraventricular septum. The intraventricular septum separates the left and right sides of the heart, who are both happy. Now we're going to add a perpendicular line. This perpendicular line helps to separate the ventricles from the atria. So what we get is a simple diagram of the heart with 2 top chambers, the atria, and 2 lower chambers, the ventricles. So now get out your pen, make it a happy pen, and let's label the left and right atrium and the left and right ventricles. You may not have room to write ventricle or atrium, so you can just abbreviate LA for left atrium and RA for right atrium. So now that we have this sketch of our heart, we're now going to add some of the blood vessels that comprise both the pulmonary and the systemic circuits. Let's start with the pulmonary circuit. The first of these vessels is the pulmonary artery. The pulmonary artery leads from the right ventricle to the pulmonary capillaries. Remember that pulmonary means lungs, and artery is a blood vessel that leads away from the heart to the lungs. We're now going to draw our pulmonary capillaries. The capillaries are the smallest of the blood vessels, and this is where exchanges of gases like oxygen and carbon dioxide takes place. Leading away from the pulmonary capillaries is the pulmonary vein. A vein is a vessel that carries blood back to the heart, and so the pulmonary vein's job is to carry blood from the pulmonary capillaries back to the left atrium. Now let's begin to draw our systemic circuit. Remember, the systemic circuit pumps blood to the remaining tissues in the body. And the largest vessel of the systemic circuit is our aorta. The aorta is an artery. And remember, arteries carry blood away from the heart. In this case, the aorta is going to be carrying oxygen-rich blood to the systemic capillaries. The systemic capillaries are much more vast than the pulmonary capillaries. Remember, they supply every tissue in the body excluding the lungs. And so the heart has to pump very, very hard to pump this blood through the systemic capillaries. So the last vessel of the systemic circuit is called the vena cava. The vena cava is a vein, and veins carry blood back to the heart. It is a happy vein. There are actually two vena cava, a superior vena cava that drains blood from the head and neck, and an inferior vena cava that drains blood from the trunk and the thorax. So we've now completed the happy diagram of the human heart and also the blood vessels that enter and leave it. And we can now clearly see the pulmonary and systemic circuits. Okay, so I'm going to cut the Bob Ross voice here. It is important you be able to draw this diagram for the exam and also to show me how the blood flows through the heart and the blood vessels and how the pressure and the oxygen concentration changes. So we're going to do that now. Okay, so we're going to start in the right side of the heart, on the right atrium, and this is the start of the pulmonary circuit. Remember, the pulmonary circuit pumps blood to the lungs where it can be oxygenated. And so the blood comes into the pulmonary circuit from the vena cava. The vena cava is a vein, and so it's carrying blood that is low in pressure. Most veins carry low pressure blood. And the reason that it's low pressure is because as blood went through those systemic capillaries, it slows down. And so the vena cava brings back this low pressure blood that's also low in oxygen. It's low in oxygen because we've dropped off all that oxygen in the tissues of the body. So we go from the right atrium into the right ventricle. Remember, in order to go from right atrium to right ventricle, we had to go through an AV or atrioventricular valve. Which valve was it? If you said tricuspid, you're correct. Remember that tri and right both have an RN in them, and so the tricuspid valve separates the right atrium from the right ventricles. From the right ventricles, we'll then go through the pulmonic valve, which is the valve leading into the pulmonary artery or pulmonary trunk. Now, I realize that the pulmonary artery is actually on top of the heart, but I cannot draw 3D structures, so I tend to draw it with it coming out of the bottom of the heart. So the pulmonary artery is carrying oxygen-poor blood to the pulmonary capillaries in the lungs. Because it's just been compressed by the right ventricle, the pulmonary artery is actually carrying relatively high pressure blood, but in this case it's blood that is oxygen poor. And so most people tend to think that arteries carry oxygen-rich blood, and that's often the case, but here's an exception. The pulmonary artery carries oxygen-poor blood from the right ventricle to the pulmonary capillaries. And so here's just a reminder. The pressure is high because we've just compressed that blood in the right ventricle, but the oxygen is low because we dropped off all that oxygen when we went through the systemic circulation. The purpose of going to the pulmonary capillaries is to drop off this carbon dioxide and pick up more oxygen. And that's just what happens within the pulmonary capillaries. The blood in the pulmonary capillaries picks up oxygen by simple diffusion from the alveoli, and it drops off carbon dioxide by simple diffusion into the alveoli. The reason this works is that oxygen is more abundant in the alveoli of the lungs than in the pulmonary capillaries, so it goes from an area of high concentration to an area of low concentration. By the same token, blood arriving at the pulmonary capillaries is saturated with carbon dioxide, whereas there's not much CO2 within the alveoli, and so carbon dioxide tends to diffuse from the capillaries into the alveoli. The end result is that blood leaving the pulmonary capillaries is now high in oxygen and low in CO2, but it is also low in pressure. Remember that every time we send blood through a capillary bed, it slows down. And this is a function of the numbers of blood vessels that are there, as well as their small, tiny diameter. There's a lot of friction in there, a lot of force is needed to get it through those capillary beds, and so it takes a lot of the compressive force of the heart to actually force blood through this myriad of tiny, tiny blood vessels. And once we emerge from that capillary bed, the blood is often quite slow and sluggish, and that's the reason we have to take it back to the heart to be compressed again before we go to the rest of the body. And so the pulmonary vein carries this oxygen-rich, low-pressure blood back to the left atrium of the heart. From the left atrium, we're now going to go into the left ventricle, but we're going to go through a valve. Which valve is it? If you said the mitral or bicuspid valve, you are correct. The mitral valve separates the left atrium from the left ventricles. We're now in the left ventricle. Remember, the left ventricle is the most muscular chamber of the heart because it has the job of pumping the blood the furthest. It's going to pump the blood through the entire systemic circuit of the body. So the blood is pumped through the aortic semilunar valve into the aorta. Again, the aorta doesn't come out the bottom of the heart, it actually arrives from the top of the heart. But that's very difficult for me to draw. So the aorta here is shown coming out the bottom. The aorta carries oxygen-rich, high-pressure blood to the systemic capillaries. It's high pressure because it was just compressed by the left ventricle, and it's high in oxygen because that oxygen was picked up in the pulmonary capillaries. So in the systemic capillaries, we're going to drop off oxygen into the tissues and pick up carbon dioxide. Oxygen moves from the capillaries into the tissues because there's more oxygen in the capillaries than in the tissues, and so it moves from an area of high concentration to an area of low concentration. The reverse is true for carbon dioxide. There is more carbon dioxide in the tissues because of cellular metabolism, and so CO2 moves from an area of high concentration, the tissues, into the systemic capillaries. At the end of the systemic circuit, the blood pressure is going to be low because we went through a capillary bed, and the oxygen concentration is also going to be low because we just dropped off that oxygen in the systemic capillaries. CO2, on the other hand, is going to be high. So the vena cava's job is to carry this sluggish blood, which is low in oxygen and high in CO2, back to the right atrium of the heart. Remember, we have both an inferior and superior vena cava. So we've now successfully diagrammed the flow of blood through both the pulmonary and systemic circuits. I suggest you do this another 2 or 3 times in order to get it down, because again, being able to draw this picture and understand it will help you to understand so much about the human circulatory system, and more importantly, it will help you do very well on the upcoming quizzes and exams. Remember, if you send me a picture of your diagram, and if it's a good picture, I will give you 5 points on your upcoming quiz. Okay, you've now reached the end of part 1 of the lecture on the human heart and circulatory system. Before you go on to part 2, please complete the following quiz questions at the end of this presentation. You will not be scored or graded on your performance on this presentation, but if you do score less than 70% on these questions, I suggest you go back and review the lecture again before proceeding on to part 2. Okay, welcome to Zoology 142. This is part 2 of the lecture on the heart. Remember, the heart is a 4-chambered pump that pumps blood through 2 circuits, the systemic circuit, which goes to the entire body, and the pulmonary circuit, which goes to the lungs. We're going to start out today's lecture by talking about the structure of cardiac muscle and comparing it to skeletal muscle. Just like skeletal muscle, cardiac muscle is striated, meaning it has bands in it, but unlike skeletal muscle, it is not voluntary. That is, we cannot voluntarily control our heart rate. So here are some structural and functional characteristics of cardiac muscle. First of all, the fibers of cardiac muscle, the cells themselves, tend to be very short, about 50 to 100 microns in length. If you remember, skeletal muscle fibers, on the other hand, were very, very long and extended all the way from the origin to the insertion of the muscle. So if we look at a muscle like the quadriceps, those muscle fibers can be over a foot long, whereas here we're talking about very short cardiac muscle fibers that are fractions of a millimeter long. The other difference between cardiac and skeletal muscle is that skeletal muscle had many nuclei, whereas cardiac muscle usually has 1 or 2 nuclei, so it's uni or binucleate. The other thing about cardiac muscle is that it branches. It's not located in single cells that extend the length between an origin and insertion, but these cells in fact do branch off into other cells. And the cell junctions are separated by something called intercalated discs. Intercalated discs are special connections that allow for electrical communication between adjacent cardiac cells, which is very important because the heart has to contract rhythmically in order for it to pump blood efficiently. So let's take a look at some other physiological differences between skeletal muscle and cardiac muscle. The first big difference is the means of stimulation. A skeletal muscle can be stimulated to contract by motor nerve impulses from the brain or the spinal cord. On the other hand, the cardiac muscle is in fact autoarrhythmic. It does not need stimulation from motor neurons in order to contract. It contracts on its own. So you may have seen these movies where somebody reaches into somebody's chest and pulls out their heart and the heart is still beating. Well, I got to tell you, that's actually possible, and I've seen it happen with fish hearts and some hearts of some lower vertebrates. The heart has autoarrhythmic cells that will stimulate it to contract. Now, that's not to say that the heart totally contracts independently. It is sped up or slowed down by nervous input, but it does have an autoarrhythmic rate that is set by pacemaker cells within the heart itself. Another big difference between cardiac muscle and skeletal muscle is the number of muscle fibers participating in contraction. If you remember back about skeletal muscle, that normally if we're lifting a very light weight, let's say a paper clip, we really don't need all our muscle fibers to contract. In that case, we only stimulate the very smallest muscle fibers using motor neurons. And remember, a motor neuron in all the muscle fibers it controls is called a motor unit. So typically, only a few motor units would be recruited to contract for something that was very light in weight. On the other hand, if we were lifting something that was very heavy, we would stimulate more motor units, and these motor units would typically have larger diameter, stronger muscle fibers. Now, in contrast, when we look at the heart, all cardiac muscle fibers participate in all contractions. So we cannot choose whether some fibers will contract or all fibers will contract. In all circumstances, all muscle fibers will contract. This is because cells are able to communicate electrically across the intercalated discs that separate each cell. And this allows for a smooth rhythmic contraction of the heart that is going to pump blood very efficiently. Another big difference between cardiac muscle and skeletal muscle is the length of the contraction and refractory periods. Remember that the refractory period is the period after contraction during which the muscle fiber cannot contract again. For skeletal muscle, the refractory period was very brief, typically 1 to 2 milliseconds. So if we stimulated muscle fibers repeatedly with skeletal muscle, we could get something called tetanus, where wave summation happens and we get this maximal sustained contraction that enables us to lift and carry a very heavy weight. On the other hand, we don't really want tetanus in the heart because tetanus would prevent blood from pumping efficiently. So we have a very long refractory period in cardiac muscle, about 250 milliseconds, and this prevents tetanus from occurring. So the result is that cardiac muscle fibers have a fairly long contractile period, around 0.25 seconds or 250 milliseconds, and they have an equally long refractory period, and this prevents tetanus or sustained contraction. I said before that one of the big things that sets cardiac muscle apart from skeletal muscle is its ability to generate its own action potentials. We call this autorhythmicity. So cardiac muscle cells are autoarrhythmic cells because they are self-excitable. They repeatedly generate spontaneous action potentials that will trigger the heart to contract. These autoarrhythmic cells act as pacemakers to set the rhythm for the entire heart. They also form the conduction system, which is the route for propagating action potentials throughout the heart muscle. There are two important collections of these autorhythmic cells in the heart. The first of these are located in the sinoatrial node. The sinoatrial node is located in the wall of the right atrium, just inferior to the entrance of the vena cava. And this is the primary pacemaker of the heart because it has the fastest depolarization rate, typically somewhere between 75 and 100 depolarizations per minute. On the other hand, the atrioventricular node is a secondary pacemaker. The AV node is located in the inferior portion of the interatrial septum, immediately above the tricuspid valve. The AV node typically depolarizes somewhere between 50 and 60 times per minute. And it is the depolarization of the AV node that will eventually cause contraction of the ventricles, whereas the depolarization of the SA node causes contraction of the atria. So even though these are the two main pacemakers of the heart, I should also mention something called ectopic pacemakers. Ectopic means out of place, and ectopic pacemakers occur when one of the other pacemakers isn't working properly. This can happen, for example, during a myocardial infarction when the heart is robbed of oxygen due to a blockage in a coronary blood vessel. And this can cause an irregular contractile force across the heart, sometimes seen as ventricular fibrillation, which we'll talk about in a few more slides. So the big picture here is that there's two different pacemaker nodes of the heart, the SA node and the AV node. Both of these nodes have an intrinsic firing rate. The SA node fires spontaneously at a rate of about 90 to 100 times per minute, whereas the AV node fires spontaneously at a rate of 40 to 50 times per minute. Now, when everything's working properly, the fastest node will set the pace of the heart. So in this case, it will be the SA node. But if the SA node is disabled for some reason, the AV node will take up its role as the primary pacemaker, but it will only depolarize or cause contractions at a maximum of 40 to 50 times per minute, which is usually not enough to sustain life. If, on the other hand, both nodes are suppressed, fibers elsewhere in the heart can fire at a rate of 20 to 40 times per minute. Again, this tends to be below the level at which we can sustain life. So if you have a disabled SA or AV node, chances are you'll need an artificial pacemaker. Artificial pacemakers are electrical devices which are implanted near the heart, and they stimulate it to contract at an acceptable rate. You've probably also noticed at some time or another that your heart skips a beat or has extra contractions. These extra contractions come from something called ectopic pacemakers, and these are tissues throughout the heart that are stimulated to contract through adrenaline or caffeine or nicotine. So if you've been up late studying for an exam, smoking like a chimney, and drinking lots of caffeine, you may notice a few of these extra beats which arise from something called ectopic pacemakers. So this slide gives us an overview of the placement of our two pacemakers as well as other conductive tissue within the heart. We're going to go through each of these in some detail. Remember that under normal circumstances, the primary pacemaker of the heart is the SA or sinoatrial node. The sinoatrial node is located just inferior to the entrance of the vena cava in the right atrium. The SA node depolarizes at a rate somewhere between 75 and 100 beats per minute, and because it's the fastest, it tends to be the major pacemaker for the heart. In addition to setting the pace of the heart, the depolarization of the SA node causes contraction of the atria, and this atrial contraction will help to eject blood from the atria into the ventricles. After the SA node depolarizes, those electrical impulses will very quickly reach the AV node. Now, the AV or atrioventricular node is located in the inferior portion of the interatrial septum immediately above the tricuspid valve. Upon reaching the AV node, these depolarizations will actually pause for about 100 milliseconds before proceeding elsewhere to the ventricles. And this is important because this delay allows the atria to contract fully before that electrical signal is sent to the ventricles telling them to contract. Imagine what would happen if both the atria and ventricles contracted simultaneously. We would basically be trying to shoot blood in opposite directions, and this would drastically affect the efficiency of the heart. So under normal circumstances, the atria contract, there's a pause, and then the ventricles would contract. And that's because of this delay that occurs here at the AV node. Once the impulses leave the AV node, it will then move to the atrioventricular bundle, also called the bundle of His. The AV bundle is the only electrical connection between the atria and the ventricles. At this point, the electrical signal will then bifurcate or split and travel down the left and right bundle branches, which course along the interventricular septum towards the heart apex. The impulses will then proceed from the left and right bundle branches to the Purkinje fibers, which transmit the impulses to the heart apex and to the superficial walls of the ventricles. Here it will stimulate the cells of the ventricles to begin contraction, and this contraction will begin at the heart's apex and move superiorly. The total time between initiation of an impulse by the SA node and depolarization of the rest of the heart is approximately 0.22 seconds or 220 milliseconds. Okay, now that we've learned about the conductive system of the heart, we're going to discuss the patterns of electrical activity produced by the heart. To do this, we have to use something called an EKG or electrocardiograph. The K in here comes from the German cardio, and we tend to use it over ECG because an ECG can also be mistaken for something called an electroencephalograph, which is used on the brain. So an EKG or an electrocardiograph measures the electrical activity of the heart via sensors that are placed on the chest and elsewhere in the body. You've probably seen an EKG in use at the hospital, or at the very least on one of those sensational medical dramas like ER or Grey's Anatomy. So the sensors on this EKG pick up the minute electrical impulses which are generated by the heart as it depolarizes. It prints these impulses on a graph or depicts them on the screen, like you can see in part A of this slide. The graphical depiction of this electrical activity is called an electrocardiogram. So the graph is the instrument used to generate the gram. And we can use this electrocardiogram to see if the electrical system of the heart is working properly. We can also use it to estimate heart rate. So in the next few slides, we're going to take a look at the different waves, segments, and intervals that are present on an electrocardiogram. Okay, the first wave we're going to look at is the P wave. The P wave is caused by firing of our SA or sinoatrial node, and it causes atrial depolarization. So the P wave is a very short wave. It lasts typically 0.08 seconds, and it's important to realize that this depolarization of the atria is not completely synced up with the contraction of the atria. If you look at the blue bar at the top of the screen, you can see when the contraction begins. And so contraction of the atria actually begins about midway through the P wave. In addition to waves, we also have something called segments and intervals. Segments are spaces between waves, whereas intervals include the wave and the adjacent segment. So the PR interval is the time from atrial stimulation until ventricular stimulation, and typically the PR interval is about 0.16 seconds. We could, however, see it get longer if there was a disruption, let's say in the transmission between the SA and AV nodes. Our next wave is the QRS complex, which represents ventricular depolarization. This is the largest and easiest to identify wave on electrocardiograms. The QRS complex is typically 0.08 seconds in duration, and remember there's going to be a delay between the actual depolarization and when contraction occurs. So if you look at the orange bar at the top of the screen, that shows the total period of ventricular contraction. So there's a slight delay between the actual depolarization and contraction. Next is the ST segment. Remember that segments do not include waves, so the ST segment goes from S to the beginning of the T wave. And this is the time when the ventricular myocytes are completely depolarized, so that depolarization started with the QRS wave and continued through the ST segment. And the ST segment is typically around 0.38 seconds. Our next wave is the T wave, which represents ventricular repolarization. Remember, with nervous tissue and with muscle tissue, it depolarizes and then it must also repolarize. So the T wave, which is about 0.16 seconds, represents this ventricular repolarization. At this point, you might be asking yourself, well, where is the wave that represents atrial repolarization? Well, atrial repolarization does happen, but unfortunately it happens at the same time as ventricular depolarization, that is during the QRS wave. And because the QRS wave is so big, it tends to block out the minute amount of electrical activity that occurs during atrial repolarization. And finally, the time between heartbeats is called a TP interval. The TP interval is variable in duration. For example, if you're just sitting here in class and your heart is beating about 60 beats per minute, the TP interval could be up to a second. Whereas if you are going to the gym and your heart rate is really racing because you're on an exercise bike, you would expect the TP interval to be much shorter because there are more heartbeats in a given period of time. So as I said before, we can use the electrocardiograms that result from our electrocardiograph study to look at the heart rate and also to determine whether all parts of the electrical conduction system are working properly. So in this slide, you can see an example of a normal electrocardiogram. Here we have a P wave, a QRS wave, and then a T wave. A little break, a P wave, a QRS wave, and a T wave. And so this would be a normal electrocardiogram, and it's showing that impulses are originating at the SA node, which causes the characteristic P wave. On the other hand, if the SA node is damaged, we might see something called a junctional rhythm. And this is an electrocardiogram where we don't always see a P wave. And that is because the P wave is missing, because the SA node is not firing, and so as a result, the AV node is going to take up its role as primary pacemaker. And so we're still going to see our QRS wave and our T wave, which indicate ventricular depolarization and repolarization, but we're not going to see our P wave all the time. And this is dangerous because without atrial contraction, the ventricles may not fill completely. In addition, it's important to realize that the AV node fires at a rate that's much slower than the SA node. As a result, the heart rate tends to go down dramatically, and we get something called bradycardia, which we'll talk about in just a minute. Another common disorder of the electrical conduction system of the heart is something called second-degree heart block, and this happens when electrical impulses are not transmitted efficiently from the SA to the AV nodes. As a result, we don't get a QRS wave for every P wave. So when there are absent QRS impulses, that indicates failure of the SA node to communicate with the AV node. So if you had a patient with either heart block or a junctional rhythm, the doctor would likely request a cardiology consult to see if the patient would be a candidate for an external pacemaker. An external pacemaker can compensate for defects in the conduction system of the heart by periodically sending electrical impulses to certain regions of the heart in order to ensure efficient contraction and pumping of blood. So heart block is an example of an arrhythmia. An arrhythmia or dysrhythmia is an irregularity in the heart rhythm, oftentimes but not always resulting from a defect in the conduction system of the heart. And here are some other examples of arrhythmias. The first of these is something called bradycardia. Bradycardia means slower than normal heart rate. If you remember that the normal heart rate for a resting person is somewhere between 60 and 80 beats per minute. If we take a look at the electrocardiogram and look at the number of big boxes between adjacent QRS waves, the big boxes here represent 0.2 seconds apiece. So if we count on there, we get 1, 2, 3, 4, 5, 6, almost 6 big boxes between one QRS and the other QRS. So basically, these heartbeats are greater than 1.2 seconds apart, and if you do the math on that, you'll figure out that gives you a heart rate of around 50 beats per minute. And this heart rate is usually insufficient to sustain oxygenation of the body, and so we call it bradycardia. On the other hand, the middle electrocardiogram shows an example of tachycardia. Tachycardia is a heart rate that's higher than normal. Normally we consider something tachycardia if it's over 100 beats per minute while the person is resting. So if you take a look at this one, you'll see that the QRS complexes are about 2 large boxes apart, or 0.4 seconds. If you do the math on that one and divide 60 divided by 0.4, you're going to come up with a heart rate somewhere between 140 and 150 beats per minute, and this is definitely tachycardia. And finally, the bottom figure shows the most serious arrhythmia called ventricular fibrillation. Here you cannot see any P waves, QRS waves, or T waves, just a big jumble of electrical activity. And ventricular fibrillation represents an unorganized electrical activity across the heart, which also results in unorganized contraction of the heart. And so a heart that's in fibrillation probably isn't pumping any blood whatsoever, and so that person is in imminent danger of death unless we correct it quickly. And usually the way that we correct ventricular fibrillations is to administer an electrical shock using an external automatic defibrillator. So now that we've talked about the electrical activity in the heart, we're going to now move on to the mechanical activity that happens during each heartbeat. Before we do this, I should talk about the technical terms we use for contraction and relaxation of the heart. So the term we use for contraction of the heart is systole. Systole is the period when a heart chamber is contracting and forcefully ejecting blood. On the other hand, relaxation of a heart chamber is called diastole. During diastole, the chamber is relaxing and expanding and usually filling with blood. So during systole, blood is ejected, whereas during diastole, the chamber fills with blood. And it's important to realize that the chambers don't all go through systole and diastole at the same time. The atria will contract or go through systole first, followed by contraction of the ventricles. Although this is the first time I've mentioned systole and diastole in this lecture, I'm guessing you probably think these words sound familiar, and that's because the words systolic and diastolic come from these words. Systolic blood pressure is the pressure when the heart is actively contracting, whereas diastolic blood pressure is the pressure in the peripheral blood vessels when the heart is relaxing. So systolic blood pressure tends to be higher, around 120 millimeters of mercury, than diastolic blood pressure, which is usually around 80 millimeters of mercury. We'll talk more about blood pressure in the next chapter. Okay, now we're going to take a look at the mechanical events in the heart happening during contraction or systole and relaxation or diastole. We're going to start out with the ventricles and diastole. So the ventricles are relaxing and they've already filled with the majority of blood that they're going to pump in the next beat. And in order to top off the ventricles, we must first have atrial systole, that is contraction of the atria. And on the EKG, this corresponds to the PQ interval.
And here the atria are contracting and filling the ventricles with an additional 10 to 20% of blood. Because the atria are contracting, the pressure in the atria is greater than the pressure in the ventricles. As a result, the AV valves will be forced open. On the other hand, the semilunar valves that go from the ventricles to the pulmonary trunk and aorta will remain closed. And during this period, approximately 20 to 25 mils of blood will be ejected from the atria into the ventricles. Again, this amount of blood is really not very large compared to the amount of blood already residing within the ventricles, which is around 50 mils. After the atria have contracted, we'll then go into a period of ventricular systole. This is the period when the ventricles themselves begin to contract. On the ECG, this corresponds to the ST interval. Remember, the QRS wave was the stimulus for the ventricles to contract, but the actual contraction goes from S to T. There are two phases of ventricular systole. The first of these is called isovolumetric contraction, and it lasts about 0.5 seconds. So isovolumetric means same volume. And during isovolumetric contraction, the ventricular pressure is rising as the ventricles contract, but it's still less than the pressure in the aorta and the pulmonary trunk, which are closed. Here, the AV valves are also closed because the pressure in the ventricles is now greater than the pressure in the atria, so both the bicuspid and tricuspid valves have snapped shut. The next phase of ventricular systole is called ventricular ejection, and this lasts about 1/4 of 1 second. Here, the ventricular pressure has now exceeded the pressure of the aorta and the pulmonary trunk. As a result, the semilunar valves will open up. The AV valves, however, are still closed. And during this period, approximately 70 milliliters of blood will be injected from the ventricles into the aorta and the pulmonary trunk. Once the ventricles have fully contracted, they will then begin to relax or undergo diastole. The first stage of ventricular diastole is something called isovolumetric relaxation. Remember, isovolumetric means same volume. And on the ECG, this corresponds to the TP interval. Here the ventricles are relaxing and the ventricular pressure is dropping rapidly until the semilunar valves will be pulled shut, causing a brief rise in blood pressure called the dicrotic notch. As the ventricles continue to relax and expand, they will then begin to suck blood from the atria. This is called ventricular filling. And this happens when ventricular pressure is less than atrial pressure, and literally it creates a suction-like force which draws the majority of the blood from the atria into the ventricles. And so we've now completed one complete cardiac cycle. Remember, the next stage, if we were to repeat this again, would be atrial contraction, which is going to give us an additional 20 to 25 milliliters of blood to put into our ventricles so that they will be able to expel that during the next contraction. Okay, so we said previously that you need a complicated device called an electrocardiogram in order to look at the electrical activity of the heart. But that's not true in order to hear the mechanical activity of the heart. You can actually listen to the mechanical activity of the heart using a simple device called a stethoscope. And you've probably seen a stethoscope before and probably even used one. And so we can use this stethoscope to auscultate or listen to various heart sounds. And as far as this class is concerned, the main sounds we're listening to is the opening and closing of the valves of the heart. Remember, there's 4 valves in the heart, the 2 atrioventricular valves, as well as the 2 semilunar valves. And so depending on which area of the chest you listen to, you can hear more of one valve than the other. Now, for students just starting anatomy and physiology, we don't expect you to be able to distinguish the difference between a tricuspid valve and a bicuspid valve, or an aortic semilunar valve from a pulmonary semilunar valve, but we do expect you to be able to tell the difference between the 2 major heart sounds. And these 2 heart sounds are called S1 and S2. The sound S1 corresponds to the closure of the atrioventricular valves when the ventricles are contracting. So during ventricular systole, the ventricles will forcefully contract and they will send blood up towards the atria, and this will snap the AV valve shut, and this can be heard very clearly on a stethoscope. The second sound, S2, corresponds to closure of the semilunar valves, and this happens when the heart begins to relax after its initial contraction. So it will contract, causing S1, or closure of the AV valves, and then as it relaxes, it will create a suction which snaps back the SL valves, causing sound S2. So hopefully in the next couple weeks in lab, you'll get a chance to try out a stethoscope and listen to these two different heart sounds. In addition to using a stethoscope to hear the sounds S1 and S2, we can also use this stethoscope to estimate the apical pulse or heart rate. The heart rate is the number of beats that the heart is making in 1 minute time. And so you can simply just place your stethoscope over the appropriate area and count for an entire minute, but this can be somewhat tedious. So many medical professionals will simply listen to the chest for about 15 seconds and take the number of heartbeats and multiply by 4. For example, in 15 seconds, if you hear 20 heartbeats, you know that you have a patient with a pulse of 80 beats per minute. Now, one of the things you'll probably notice after listening to the heart rates of several people is the heart rate can speed up and slow down as people breathe in and out. And this process is called normal sinus arrhythmia. And as the name implies, it's completely normal in people. So normally, as you exhale, your heart rate will decrease. And as you inhale, the heart rate will increase dramatically. So you can have a heart rate that's in the 80s and then take a deep exhalation and drop it down to 65 perhaps. And so normal sinus arrhythmia is a normal finding in healthy human beings. Before we go on to talk more about the cardiac cycle, we need to go through some vocabulary. Remember that the TP interval was the interval between one heartbeat and the next, and it tends to decrease as the heart rate increases. And so at a heart rate of 75 beats per minute, the TP interval will be somewhere around 0.8 seconds. We now need to talk about something called end diastolic volume. The end diastolic volume, or EDV, is the volume of blood in the ventricles at the end of diastole, and is typically around 130 milliliters. In contrast, the end systolic volume, or ESV, is the volume remaining in the ventricle at the end of ventricular systole, and it's typically around 60 milliliters. By subtracting the end systolic volume from the end diastolic volume, you can get an idea of actually how much blood is ejected for each contraction of the ventricles. So if the end diastolic volume is around 130 milliliters and the end systolic volume is around 60 milliliters, this should give you a stroke volume of around 70 milliliters. And so the stroke volume is the volume of blood ejected from the ventricles with each beat of the heart. And again, here's the equation you would use to calculate stroke volume. Stroke volume is end diastolic volume minus end systolic volume. And yes, you will need to be able to calculate this for the exam. Now, I also need to mention something about the pressure of blood in the major blood vessels leaving the heart. You probably had your blood pressure taken before using something called a sphygmomanometer, which is just a fancy word meaning blood pressure cuff. And the sphygmomanometer measures blood pressure at the peripheral blood vessels, for example, in the brachial blood vessels in your arm. But here we're going to talk about the blood pressure and the major vessels leaving the heart. For example, the aorta has a blood pressure of around 120 millimeters of mercury, whereas the pulmonary trunk or pulmonary arteries only have a blood pressure around 30 millimeters of mercury. Why the big difference? Well, if you remember, the pulmonary trunk and pulmonary arteries only have to pump blood through the capillaries of the lungs, and so we need a lot less pressure to do this because the capillary bed isn't as large as the systemic circuit. On the other hand, the systemic circuit includes all the other capillary beds in the body, and it's much more vast than that of the pulmonary circuit, so we have to have a higher pressure in order to get this blood all the way through these capillaries. You should also remember that there's a big difference in the thickness of the ventricular walls. The right ventricle, which pumps to the pulmonary circuit, is relatively thin, whereas the walls of the left ventricle is very, very thick because it has to do a lot more pumping. So even though there's a big difference in the thickness of the ventricles and also the pressure of the blood within the pulmonary trunk and the aorta, the volume ejected by each of these ventricles is the same, usually around 70 milliliters. So here's a question for you. Why does the stroke volume need to be the same for the left and right ventricles? Be thinking about this because we're going to come back to it in just a few more slides. Our next vocabulary term is cardiac output, and cardiac output is the volume of blood ejected from the ventricle every minute. So cardiac output equals the stroke volume, which is usually 70 milliliters, multiplied by the heart rate. So if you had a person with a heart rate of 100 beats per minute, their cardiac output would be 7,000 milliliters or 7 liters. So here's a question for you. What is the cardiac output for a person with a heart rate of 120 beats per minute and a stroke volume of 70? To figure this out, you simply multiply one times the other. So 120 heartbeats per minute times 70 mils is around 8,400 milliliters per minute, or 8.4 liters per minute. Another term we need to know is something called the cardiac reserve. The cardiac reserve is the ratio between the maximum cardiac output a person can achieve and the cardiac output at rest. So think about what your cardiac output would be at rest. What is a good resting heart rate? So if you're like me, your resting heart rate is probably around 80 beats per minute. So at 80 beats per minute, your cardiac output could be calculated by 80 times 70. And that should give you a cardiac output of around 5.6 liters per minute. Now, think about what your maximal heart rate is. Let's say you've gone to the gym and your heart rate's gone up to 180 milliliters per minute. If we multiply 180 times 70 milliliters, we get something like 12.6 liters per minute. So if you subtract the first measurement from 12.6, you'll get an idea of what your cardiac reserve is. And the more healthy you are and the more athletically trained, the greater the cardiac reserve you'll have. So you should remember from the last slide that cardiac output can be determined by multiplying the heart rate times the stroke volume. And so the body can increase cardiac output simply by increasing the heart rate. But it also has other methods for adjusting cardiac output by adjusting the stroke volume. And there are three factors that affect stroke volume, and these are preload, contractility, and afterload, and we're going to talk about each of these in some detail. And the first of these factors is preload. Preload is determined by the degree of muscle fiber stretch within a chamber of the heart. And the concept of preload is described in something called the Frank-Starling law of the heart. That is, the greater the starting volume of blood in a heart chamber, called the end diastolic volume, the greater the degree of muscle stretch. And with more muscle stretch, we will have increased contraction of the heart, ejecting a greater than normal quantity of blood. So let's look at an example. Imagine that we have a greater than normal volume of blood returning to the right atrium via the vena cava. This greater than normal amount of venous return will cause the right atrium to become distended or enlarged above normal, and this greater enlargement will stretch the cardiac muscle cells within the atrium. But because of preload, when the cardiac muscle cells in the atrium contract, they will contract more forcefully because they were stretched. And this more forceful contraction will help to eject a greater quantity of blood than normal. So basically, preload is a way in which the heart can catch up if venous return is greater than normal. And this doesn't just hold true for the atria, it also holds true for the ventricles. If the ventricles are filled with a greater than normal quantity of blood, they will contract more forcefully to push that excess along to the next blood vessel or next heart chamber. Another factor that can increase stroke volume during exercise or other periods of increased cardiovascular demand is something called contractility. And this is independent of muscle stretch. It has nothing to do with muscle stretch. But here the contractile strength of the heart increases in response to certain chemicals such as epinephrine and norepinephrine. So when you're exercising, epinephrine is released from the adrenal medulla and causes an increased contractility of these chambers, forcing more blood out than normal so we can keep up with increased cardiovascular demand. Substances which increase the contractile force of the heart are called positive inotropic agents, and examples include epinephrine, thyroxine, and also the drug digitalis. So the presence of positive inotropic agents in the bloodstream will cause a complementary increase in the contractile force of the heart. And just like the concept of preload, this results in a greater amount of blood ejected from all the heart chambers during contraction, which also results in a lower than normal end systolic volume. The next factor affecting stroke volume is something called afterload. And afterload is caused by back pressure exerted by arterial blood in the aorta and pulmonary trunk. And this one can actually reduce stroke volume, whereas the first two we talked about, preload and contractility, actually boosted stroke volume. Afterload can actually reduce stroke volume if blood pressure in the aorta or pulmonary trunk is too high. In healthy people, the blood pressure within the aorta is around 80 millimeters of mercury, whereas the blood pressure in the pulmonary trunk is around 10 millimeters of mercury, and this does not have a large effect on determining stroke volume. On the other hand, if you have somebody that is hypertensive, they have a greater than normal blood pressure, and the blood pressures, especially in these blood vessels, are higher than normal, this can cause an increased amount of back pressure on the ventricles. Basically, the ventricles have to expel a lot of their contractile force just to overcome the greater than normal pressure in the aorta and pulmonary trunk. As a result, they don't expel as much blood as normal. And so afterload, if it increases, will actually reduce stroke volume and reduce cardiac output. Now, we said before that the heart is unique in that it is autorhythmic. That is, it has nodes within it that will set its internal heart rate. And the most important of these nodes is the sinoatrial node, which has a normal firing rate of about 90 to 100 beats per minute. Now, this rate is actually modulated by input from the brain, principally the medulla oblongata. And we can accelerate this rate or we can slow it down. So sympathetic stimulation is going to tend to speed up heart rate and also increase the force of contractility. So that's going to inject more blood at a faster rate. On the other hand, parasympathetic stimulation, as from the vagus nerve, is going to slow down heart rate. So think about what happens to your heart rate when you're at rest and you're listening to some soft, soothing music. Your parasympathetic nervous system will actually depress heart rate from its normal rate at about 100 beats per minute all the way down to about 60 to 70 beats per minute. On the other hand, if you are very excited or you're exercising, sympathetic nerve impulses will tell your heart rate to speed up and will also tell it to contract more forcefully. So this slide is just a recap or review of what we talked about previously, that is the chemicals that can affect heart rate and also contractility. So the catecholamines are drugs like epinephrine and norepinephrine, and they tend to increase heart rate and also increase the force of contraction. Whereas the neurotransmitter acetylcholine, which is released by parasympathetic nerves, mimics parasympathetic effects. That is, it tends to reduce heart rate but has no effect on contractility. So here are examples of 3 drugs that are commonly used in hospitals to modify the contractility and also the heart rate. The first of these is epinephrine. Epinephrine is a catecholamine drug. It stimulates an increase in heart rate, and it also stimulates the contraction of the heart. It tells it to contract more forcefully. And so we typically use epinephrine on a patient that's asystolic, and we use that to hopefully get the heart to start beating again so that we can achieve a normal sinus rhythm. On the other hand, the drugs glycopyrrolate and atropine sulfate are not used to restart the heart, but can be used to reduce bradycardia. Oftentimes what will happen to somebody in surgery is that their heart rate may go down because of the administration of opioid anesthetics. And if it goes down too low, we're worried about bradycardia and compromised cardiac output. And so we can give atropine sulfate or glycopyrrolate to raise the heart rate back up to acceptable levels. Okay, before we wrap up our lecture on the heart, we're going to talk about some disorders that are associated with the cardiovascular system. The first of these has to do with plasma lipid levels. If you remember back to the first lecture, we talked about coronary artery disease and said that it was a major risk factor for heart attacks or myocardial infarctions. Well, the primary cause for coronary artery disease is elevated levels of LDL lipids within the bloodstream. LDL stands for low-density lipoproteins, and these are proteins that are carrying high concentrations of fat and triglyceride throughout the body. On the other hand, HDLs are high-density lipoproteins, and their job is to scavenge low-density lipoproteins from other places in the body and transport them to the liver where they can be gotten rid of. And so the HDL is basically your good cholesterol, whereas your LDL is your bad cholesterol. And so you want to avoid getting too much LDLs in your diet, so avoid foods like eating too many eggs in one day or eating high cholesterol foods like shellfish every day. On the other hand, you can boost your HDLs by simply exercising, eating green leafy vegetables, and also refraining from smoking. Now, these guidelines won't guarantee that you have acceptable blood triglyceride levels. For example, I used to know a guy that was my advisor in grad school, and he had very high cholesterol. He was a very athletic guy, and he didn't eat very much fat to begin with, but after his cholesterol numbers came in, his wife put him on a very, very strict diet. Basically, I would watch him in these faculty meetings, and he was eating what looked to me like twigs and bark and things like that, and his LDL numbers didn't really budge, and so he was still at risk for coronary artery disease. And that's because that even though food is one source for LDLs, the other source is our liver. Your liver can actually synthesize low-density lipoproteins from other food sources. And so if you have bad genetics on your side, it doesn't really matter what you do in your diet, your LDLs can still be elevated. So one way that we can treat people with higher than normal levels of LDL cholesterol in their blood serum is to give them a statin drug, for example, Lipitor. And Lipitor helps to precipitously reduce the amount of circulating triglyceride in the body. Unfortunately, it doesn't actually reduce the size of the arterial plaques very much, so once they're there, they don't tend to go away, but it does help to stabilize the plaques. So really the best bet is to follow your mother's advice, that is, an ounce of prevention is worth a pound of cure. And in order to reduce your chances of getting coronary artery disease and later on getting a heart attack, you should have your cholesterol checked regularly. You should also exercise regularly, avoid smoking. You should eat plenty of unsaturated fats such as fish oils and peanut oils, and reduce your intake of saturated fats. So we're talking about things like cheese and butter and other animal fats. So now we're going to look at some structural defects that sometimes occur in the heart, and these are congenital, meaning they're present at birth. And the first one of these is something called a patent ductus arteriosus. Now this happens when the transition from the fetal period to the neonatal period isn't made smoothly. Normally during the fetal period, we're not sending blood to the pulmonary circuit, or we're not sending very much blood. And the reason is because the baby is not breathing in utero, that is, the oxygen is coming from the mother's blood. So there's a diversion or shunt between the aorta and the pulmonary artery that helps to bypass the pulmonary circuit. Normally, this shunt will close up very soon after birth. But if it does not, we can end up with something called a patent or open ductus arteriosus, in which we have mixing of oxygen-rich blood from the aorta with oxygen-poor blood from the pulmonary trunk. And this can make circulation within the heart and circulatory system very inefficient. And so if somebody has a patent ductus arteriosus, they will usually need it to be repaired surgically. Another congenital defect is something called a patent foramen-ovale. Remember, patent means open, and foramen-ovale is literally a hole in between the left and right atria. And again, this is something that is normal to have during the fetal period because it helps to divert blood away from the pulmonary circuit and send it directly into the systemic circuit. And that's because we're not oxygenating blood within the lungs of the fetus. However, soon after birth, a flap should close over the foramen-ovale, and this flap will eventually heal into the side of the heart, becoming a depression called the fossa ovalis. If it does not happen, you will then have a neonate that actually has a hole in their heart, and the same thing will happen that we saw in the previous slide. We'll have mixing of oxygen-rich and oxygen-poor blood, which makes the cardiovascular system very inefficient. And so in these cases, we will have to go in and surgically correct this defect in the heart wall. Another disorder affecting the heart is something called congestive heart failure. Congestive heart failure is a chronic or acute state which results when the heart is not capable of keeping up with venous return, and it results in an accumulation of fluids within the pulmonary or systemic circuits. It also results in hypertrophy of the ventricles. Hypertrophy means greater than normal growth. So at left you can see a normal heart, and on the right you can see a congestive heart. How do they look different? Well, the walls of the congestive heart are much thicker, and you would think that having more muscle in your heart would be a good thing, but it's actually not. It actually reduces the efficiency with which the heart can pump. And so a congestive heart can't pump as much for every beat as a normal heart can. So anytime you see a patient with an enlarged heart, you may want to worry about their propensity for congestive heart failure. So there can be several causes for CHF. These include coronary artery disease, hypertension, previous myocardial infarction, valvular disorders, or congenital defects. So if you remember to a few slides back, I asked you what would happen if the left and right ventricles did not pump the same amount of blood. Well, basically the answer is congestive heart failure. The symptoms of congestive heart failure will vary depending on which of the two ventricles is most affected. In left-sided heart failure, the left ventricle is damaged and doesn't pump as much as it should. Remember that a normal stroke volume for the left ventricle should be around 70 milliliters per beat. Let's say, however, that this heart is damaged or hypertrophic, and so now it's only pumping 50. As a result, blood will back up into the lungs, resulting in pulmonary edema, which can cause suffocation and lack of oxygen to the tissues. So somebody that has left-sided failure will always have this persistent cough, especially in the evenings, and it's resulting because they have a greater than normal volume of blood backing up in the pulmonary circuit because their systemic circuit, that is their left ventricle, isn't pumping properly. On the other hand, if you have failure of the right side of the heart, we're going to see a backup of blood within the systemic capillaries, and that's going to show itself as in peripheral edema. That is, we're going to see people that have ankles that are swollen or feet that are swollen, and it's because there's a greater than normal blood in this area, and some of that blood plasma is being forced out of the blood vessels and causing a greater than normal volume of interstitial fluid, resulting in swelling or edema of the appendages.